participant of the day, Mr. Sheps King McAlpin. And he will be presenting on the effects of the aminocaproic acid and a protein on myotube development in fascicle-like constructs. And hopefully I got that correct, being a psychologist. But anyway, please give a round of applause to Mr. McAlpin.
Good afternoon. Testing. Testing. Good afternoon, ladies and gentlemen. My name is Sheps King McAlpin. I'm a senior biology major and a mathematics minor here at Morehouse College. Today I'm going to be talking to you all about the effects of aminocaproic acid and a protein on myotube development in fascicle-like constructs. Very lengthy title, but bear with me, I'll simplify. So for a project overview, EBICS. EBICS stands for Emergent Behaviors of Integrated Cellular Systems. EBICS is a National Science Foundation funded science and technology center. It's a collaboration of scientists with the goals of creating biological machines. An example of a biological machine would be an organism that swims through a body of water, and when it detects an oil spill, enzymatically degrades it. Another example of a biological machine would be an organ that serves as an insulin pump for diabetic patients. The idea is that skeletal muscle will act as the actuators or the movers of these organic machines. One of the projects in the lab that I worked in is to recreate the 3D hierarchy of skeletal muscle tissue. So we know that skeletal muscle tissue functions in an organized manner. We want to recreate this so it can function in the organic machine. My project was to optimize growth conditions for the muscle cells in these engineered muscle tissue. The level of tissue that we decided to engineer was the fascicle. The fascicle is a bundle of muscle cells with a high aspect ratio. By high aspect ratio, I mean that one dimension is very large compared to the other dimension. So in this case, a fascicle is very long compared to how wide it is in diameter. Fascicles range from 50 to 500 microns in diameter. Now I'm going to give you a step-by-step method of exactly how we went about engineering these fascicle-like constructs. We started off with an iron mold with 350 micron holes. We inserted 350 micron pins. And we poured in PDMS. PDMS is polydimethylsiloxane. It's a biologically compatible organic polymer. So it starts off as a liquid, and then when you heat it, it polymerizes and hardens. It has a rubber texture. So we cured or heated the PDMS in the oven. We removed the pins, leaving holes in the PDMS. We removed the PDMS slab from the mold. Next, we cut the slab up into small rectangles. Each individual rectangle, we inserted 3 holes, punched 3 5mm holes. Next, we plasma bonded this PDMS chip to a glass cover slip. Then we reinserted pins in the outer wells. Next, we placed gelatin inside of these outer wells. We cured the gelatin, or heated the gelatin in the oven. This actually allowed the gelatin to harden. Then we removed the pins. This left holes in the outer wells again. Next, we inserted a mixture of C2C12, mouse myoblast, and fibrin hydrogel. Fibrin is a fibrous protein that the cells use to form muscle strips. And a myoblast is a stem cell precursor of a muscle cell. The middle well was used as a medium reservoir. So medium is the nutrients for these cells in these muscle strips. The media actually overflows from this well and falls into these outer wells. You don't want to stick anything in these wells to disturb these developing muscle cells. The gelatin that we placed in here earlier actually degrades after a couple of days. And this leaves free-floating muscle strips in the outer wells. So you end up with what we call a fascicle device. It's a 3-well PDMS chip bonded to a glass cover slip. Again, the myoblast is seated in a fibrin hydrogel. The fascicle-like constructs are on the order of 5 millimeters in length and 100 to 300 microns in diameter. This image right here represents the development of the myoblast after seeding. As you can see, in the first 3 to 5 days, the cells simply divide and proliferate. Within 2 to 3 weeks, the cells differentiate into actual muscle cells, and they fuse and form myotubes. So the size of these fascicle-like muscle constructs is limited by diffusion of nutrients. Again, these cells are fed by media. So if we were to construct very large constructs, the media couldn't reach these cells at the center of the construct, and they would die. So we're limited to the size of the construct that we can make. We're limited to a size of about 300 microns. The idea is that we'll take these 300 micron fascicle-like constructs and combine them together to scale larger muscle tissue. So what I did is actually I tested the effects of 2 chemicals on muscle development. These chemicals are aminocapric acid and a protein. These chemicals are anti-fibrinolytic agents. What this means is that they inhibit plasmin, which is an enzyme. The muscle cells that we use to seed in the devices actually naturally produce plasmin. Plasmin degrades fibrin. However, the cells need fibrin to form these fascicle-like constructs. The cells use the fibrin as a scaffold, sort of something to hold on to as they develop into myotubes. So, these chemicals actually block plasmin and leave the fibrin intact. When the fibrin is left intact, the cells can then use this fibrin to form these fascicle-like constructs. Without any anti-fibrinolytic chemical, these constructs will actually fall apart and look like this. We saw some interesting results from this experiment. We saw that myotube development, although the myotubes need the anti-fibrinolytic chemicals to develop, as you increase the concentration of either chemical, the actual development worsens. The fascicle-like constructs have a smaller diameter when growing in a protein. And a small diameter is good. A small diameter means that the construct is dense and has a lot of muscle tissue in it, rather than extracellular material, which is not really useful. We also saw that the constructs are more uniform along their length when they are grown in a protein. So we looked at muscle growth in 2D on a 24-well plate and in 3D using the fascicle device I explained earlier. We looked at construct diameter, we did a cell count, and we looked at 2D proliferation. This represents a 24-well plate with muscle cells grown on it. Each column represents cells grown under the same conditions. This image actually shows that when you increase the concentration of aminocapric acid, the myotube development actually worsens. So you see over here, there's a lot of nice long myotubes. When you get over here, there's hardly any, they're really short. This image shows it's something similar. When you increase the concentration of a protein, the myotube development worsens as well. So you can see nice long myotubes over here. When you increase to 10%, they're a lot shorter and smaller. So we also did a 2D cell count. So we counted the cells on the 24-well plates. And we did this using a DAPI nuclei stain. So we stained the nuclei of the individual cells. And we counted using software called ImageGen. So this is the image of a DAPI nuclei stained version of this. So as you can see right here, this is a myotube. These are the cells within one myotube. And again, we saw that when you increase the concentration of aminocapric acid or a protein, the actual cell count decreases, which supports the the data that I showed before this. So we also looked at cells in the actual constructs. But instead of doing a simple cell count, we looked at cell density. How many cells are in a specific area of the construct. We saw that the density is a lot higher when the cells were growing in protein rather than an aminocapric acid. Again, protein seems to be the winner in comparing these two chemicals. We looked at the diameter decrease of these constructs. Again, a decrease in diameter is important and it's good. A decrease in diameter indicates that the cells, that the constructs are highly bundled, very dense cells. So this represents a construct right after seeding, right after placing the cells into the device. This is over time, it actually decreases. So we measured how much the constructs decrease. And we see that the constructs decrease a lot more in a protein rather than an aminocapric acid. We also looked at construct uniformity. So how uniform are these constructs along their length? In aminocapric acid, we see that the constructs tend to have lumps on them, which we don't want. However, in a protein, the constructs are a lot more uniform along their length. This is good. So in conclusion, we see that a low concentration of a protein is best for myotube development in fascicle-like constructs. A protein, in low concentrations of a protein, we see better 2D proliferation, we see a higher cell count in 2D and in 3D, we see a smaller diameter, and we see that constructs are more uniform along their length. For future work, we want to look at the tension of the actual constructs. So there's a balance between having nice, densely bundled, thin constructs and the strength of these constructs. Because again, we want to use this for organic machines, so they have to be able to bear force. So they can't be too weak. So we want to measure the tension of the constructs. We want to look at different concentrations because we showed that when you increase the concentration of either chemical, the development worsens. But how low can the concentrations get? So we want to see what really is that optimum concentration of either chemical to use. And what we also want to do, which is really important, is to standardize the aminocapric acid in a protein based on their enzymatic activity. So for example, we don't know that 0.3% of a protein is necessarily better because we need to see how effective it is, how much enzyme is actually being inhibited at 0.3% a protein compared to 2 milligrams per mil of aminocapric acid. I wanted to acknowledge EBIX and NSF for funding this research. I want to acknowledge my PI, Dr. Harry Sada, and I want to acknowledge my mentor, Devin Neal. And if there are any questions, I'll take them now. Thank you.
Excellent presentation, and I thought your graphs definitely supported your conclusions. But I forgot back from the beginning, once you grow these muscle cells, what are you going to use them for?
Oh, so the growth for the muscle cells, they're used for pretty much organic machines. That's the idea behind it. That's what the funding is for. It's an NSF funded project. So yeah, so we want to make actually like robots, but instead of using inorganic material like metal, we want to use organic material like we're made. So we actually organic robots we can control. So that's the big project behind what we're trying to grow muscle tissue for. And the muscles will be actually like the movers of the robot. That's what would allow the robots to move. So that's what it's used for. But it also can be translated from the bioengineering field to the biomedical field, where you're trying to recreate functional muscle tissue that can be used to treat muscle injuries. So it kind of has that dual function. But the funding is for the bioengineering for the robots.
Good presentation. Have you thought of or know of any alternate ECM protein or even non-organic protein or substance that will hold up like fibrin, since that would degrade less?
So another protein to substitute for fibrin?
For scaffolding, besides fibrin.
maybe collagen. That's another protein that we can try. But that's been using other research.
Also, great presentation. Just a few brief questions. The time still allows. What was your basis for the concentrations that you did use?
It was based on, pretty much based on trypsin inhibitory units. So the protein was between 7 to 10 trypsin inhibitory units, but the aminocapric acid didn't actually have a sort of enzymatic, we don't know what the enzymatic activity is, not standardized, so we just started low and worked our way up. The protein is a liquid, so it's actually volume based. So we don't want the volume to be too much of the chemical, because right, this is in media, this is in media that's used to feed the cells. So we don't want the percentage to be too much of the chemical. So the highest we went is to 10%. That's a significant amount, and lowest is 0.3. So it's kind of just really general. It wasn't like we based it off of a lot.
That makes sense. But based on the biology, like you just said, and then maybe prior research, was there like a range in this field of concentrations or volume percentages?
Yes. So for aminocapric acid, those concentrations are around the same that was used in previous bioengineering papers.
Okay.
So we took that, pretty much tested what was already being used with something new.
And then my last question is it possible it could be an opportunity window? So rather than it be monotonically decreasing, could you say, maybe at low concentrations, which you've already noted that you're going to do, maybe at low concentrations, it's also bad, but then at a certain sweet spot or opportunity window, you find the level of concentration that you need to increase the diameter, to decrease the diameter or increase separation. So my question again is it possible that it might be a inverted U-shaped phenomenon, or do you think it's always going to just monotonically decrease?
No, that's a great point to bring up. And that's one of the future goals is to look at this right here, because we see that on this side it gets worse. But we would expect that there will be a certain concentration that's too small that would actually cause it to decrease on the other side. So that U-shape, we're not sure if it's actually here or maybe a little bit smaller. So that's what we're going to try it again with lower concentrations to sort of see what really is that optimal concentration that you want to use. You're welcome.
Thanks, sir. So folks, we're down to the home stretch. I just want to, first of all, at this moment, just say thank you for your time. You've been here for a good time block. That's a long time to sit on your cane, period. So you get a little glossy-eyed, you get a little itchy, twitchy, because you kind of want to move on to the next thing. Appreciate the fact that the students that we're talking about are working on their work, what they're showing you for months, dating back to last summer, this fall, and they've been working not only to do the work that they've shown you, but also to summarize it in 10 to 15 minutes. As we know, that's an effort. So just a thank you on their behalf for listening and giving them your attention. I also have to just show respect to the science folks, because I am not a science person at all. I am business. I'm 100% dollars and cents and markets and such. And he started talking about this makes no sense to me. Zero. So I appreciate the fact that y'all can even speak like this, because I cannot. I also want to acknowledge again, I want to make the point that what we're trying to do right now is celebrate innovation and the importance of idea sharing. That's the core of innovation, that we share ideas. And so that's what we're practicing right now. Not only just from business, not only just from science, but for the sake of good ideas. So as we hear them and we're inspired by them and we mesh them together, we find things we couldn't have found before. So that's the reason why we're doing what we're doing right now. Okay, so there's 3 things left. There is a spoken word artist we're bringing forward right now. There's a final business plan presenter. Then I'll conclude, and then we can move on. All right. So again, appreciate y'all hanging there. We're almost finished. I want to bring up our 3rd spoken word presenter. His name is Yulato Singh. He's an English major and he's Morehouse College, class of 2014.